This polypeptide segment contains 16 amino acid residues. If the side chains are replaced by simple spheres, we can focus on the structure of the polypeptide backbone. Each amino acid residue contributes an NH group, an alpha carbon, and a carbonyl group to the backbone. The shape of the polypeptide backbone is called its secondary structure. We can draw the electronic structure of the bonding atoms in different ways, because the electrons of the peptide bond are not just shared by the carbon and nitrogen atoms, but also spend time near the very electronegative oxygen atom. This resonance tells us that the peptide bond is somewhere between a single bond and a double bond. Because double bonds have a limited rotation, this part of the backbone is relatively inflexible. As a result, most of the rotation in the polypeptide backbone occurs at the bonds involving the alpha carbon. Limited rotation is one factor that constrains the secondary structure of the polypeptide. Another factor to consider is hydrogen bonding. Each backbone NH group is a hydrogen bond donor, and each carbonyl group is a hydrogen bond acceptor. A polypeptide chain often folds so that its hydrogen bonding groups interact with each other. This can produce two types of regular secondary structure, the alpha helix and the beta sheet. In an alpha helix, as shown here, the polypeptide backbone coils up so that the carbonyl group of one amino acid residue forms a hydrogen bond with the NH group 4 residues farther along the chain. All the hydrogen bonds have the same geometry, so the coiled shape of the alpha helix is the same, no matter which amino acids are involved. This makes alpha helices easy to recognize. When the backbone atoms are shown in space-filling form, we can see that the helix is not hollow. And if we add the complete side chains of all the amino acid residues, we can see that they point outward from the helix. It's difficult to see secondary structure when all the atoms are visible, so an alpha helix is often represented by a coiled ribbon. The other commonly occurring regular secondary structure is the beta sheet. As before, we can ignore the amino acid side chains in order to focus on the polypeptide backbone. A beta sheet takes shape when hydrogen bonds form between adjacent segments of polypeptide. Each hydrogen bonded segment is called a beta strand. Multiple beta strands are lined side by side to form a beta sheet. The beta strands do not all lie in the same plane, so the beta sheet is actually curved. In this beta sheet, the polypeptide strands are arranged to optimize the geometry of the hydrogen bonds between the backbones. We can see this more clearly by representing the polypeptide segments as flat ribbons, with an arrowhead at the end closest to the C-terminus. This structure is called an anti-parallel beta sheet because adjacent strands have the opposite, or anti-parallel, orientation. Beta sheets can also be constructed from polypeptide segments with the same orientation. Here, the strands are parallel, so their C-terminal ends point in the same direction. In a parallel beta sheet, the hydrogen bonds are somewhat distorted, so this sheet is slightly less stable than an anti-parallel beta sheet. In a beta sheet, as in an alpha helix, the side chains are not involved in forming secondary structure. We can see that the side chains project away from the backbone segments on both sides of the beta sheet. We can also see that the beta sheet is a solid structure when atoms are shown in space-filling form. Both parallel and anti-parallel beta sheets and alpha helices are called regular secondary structures because the polypeptide backbone has a repeating conformation from one residue to the next. Segments of polypeptide that do not form alpha helices or beta sheets are said to have irregular secondary structure. In these segments, each amino acid residue has its own conformation. In some proteins, the secondary structure is almost entirely irregular. Proteins can be classified by their secondary structure. Some are constructed mostly from alpha helices. Some are constructed mostly from beta sheets. Most proteins include both alpha and beta structures. Notice that where alpha helices and beta strands occur, these elements of secondary structure are connected by segments of irregular secondary structure.